All right, well, thank you so much. It's a tremendous honor to be presented with this award and to get to talk to you folks about my science. The former awardees are some of the human geneticists that I really respect the most in this field, and so it's amazing to sort of get to join the ranks and continue the tradition. I'm going to indulge a little bit and talk about my background, even though Tuli's covered some of it, and then I'm going to talk about some work that we've been doing in the areas of understanding common and complex disease mechanisms, and then shift gears and talk about some discoveries that we've made in identifying predictors of treatment outcomes. And then maybe indulge a little bit again at the end and talk about some of sort of my perspectives on where this field is going and why I think it's an extremely exciting time to be a human geneticist. So, as Tuli mentioned, I did my PhD with Itzhak Peyer at Columbia, where we worked primarily on population genetics, and specifically developed tools like this algorithm called Germline for detecting segments of identity by descent, and built and generated really great looking figures like this one, showing the relatedness, the extensive relatedness of individuals of Ashkenazi Jewish ancestry. But in fact, if you expand this out to larger cohorts, you will see these kinds of hairballs of relatedness in essentially any population. And so one of the sort of lessons learned from that time that continues to echo is that we are all much more genetically related than we think. Subsequently, I worked with Alkis Price at the School of Public Health and switched more in the direction of statistical and clinical genetics. And there, one of the sort of findings that I'm very proud of is this analysis identifying a significant proportion of common disease heritability in regions of open chromatin, highlighted by this big blue bar here. In fact, I think this sort of, again, led to a lesson of thinking in our analyses beyond just individual associations and to the genome-wide level, and to try to characterize genome-wide parameters of disease architecture that can then inform how we conduct our studies. And sort of towards the end of that, we worked on this method called the transcriptome-wide association study, or TWAS, which I'll come back to, but for integrating various molecular measurements together with disease measurements to identify individual mechanisms. So now moving from a sort of global polygenic estimates to local mechanistic estimates. And this tool, among others, is now fairly widely used for interpreting the associations that we get out of common disease studies. And there, the lesson for me at least, was thinking about the extreme, sometimes extreme, but certainly very high polygenicity of most common complex traits, the very, very large number of associations we expect to find. We need methods that can work at scale towards pointing us to the putative mechanisms. And so that all brings me to the work that I've been doing in my lab at Dana-Farber in the Harvard Medical School, and it focuses on these three areas which somewhat overlap and continue the threads that I started earlier in my career, understanding common disease mechanisms, connecting those now to the world of cancer through germline and somatic interactions, and predicting clinical outcomes in patients. And so I'll start by talking about work that we've been doing on characterizing common disease mechanisms. So the paradigm for identifying associations for common disease for now several decades has been the genome-wide association study, which essentially produces figures like this, where there are polymorphisms along the x-axis and statistical significance along the y, and a number of associated loci. And we've gotten really good at generating these kinds of figures, but what has been a lot more difficult is moving from this kind of map to the actual map or network of mechanisms that we're interested in. And this area in general, I sort of see through this cartoon of trying to connect genetic associations to put them in their place into this network so that we can actually understand the disease. Another paradigm for doing that has been the integration of other molecular data. And so if we think about a polymorphism as often acting through an expressed gene. We can identify associations between them, polymorphisms and expression through eQTL studies, and then we can work forward into the association studies. And then finally, coming back to the methods that we developed, the TWAS integrates those two components by building predictors of expression and then identifying the associations between that predicted expression and the disease in the GWAS study. Of course, there's been other approaches, including colocalization and various forms of fine mapping. Essentially, the idea is, can we look at molecular data over here and connect it to the disease data over here? Having developed some of these tools, more recently we've been thinking, again, back to this lesson of estimating genome-wide, and in particular we were really curious to understand to what extent this kind of molecular integration of expression driving or eQTL data actually explains disease mechanisms in total. And so with work that was led by Douglas Yao in my lab, we sort of envisioned this model where variants affect. Let's see if this works. Maybe not. Here we go. Yes, great. Okay, so where variants affect the phenotype directly, but can also affect the phenotype through mediating genes. And we want to understand how much of the trait heritability is explained by these mediating genic effects. And we can actually estimate that using a sort of fancy regression where we incorporate the eQTL measurements and the squared eQTL measurements and the squared disease measurements. And so when we do this, the punchline of this analysis was essentially one genome-wide number, which is that 10% of the disease heritability in these common representative phenotypes that we looked at could be sort of considered mediated by gene expression. And I think you can think of this as a sort of glass half full or glass half empty result. I thought it was great that we're able to actually explain a substantial portion using steady state expression that was not always collected for this specific task, that was mostly collected to just understand basic biology. But of course it's really important to understand where the rest of this variation could be explained. And in particular, around the time of this study, this sort of question of where the disease eQTLs or where are the missing colocalizations between expression and GWAS happening started popping up. And in case you don't believe me, there was actually a review paper with this same exact title, Where are the Disease-Associated eQTLs, a fantastic review actually discussing potential mechanisms and where they could be hiding. And so we thought about this question as well, and in particular, how other potential mechanisms fit into this molecular cascade between polymorphism, expression, and trait. And in particular, thinking about lower level epigenetic and regulatory features that could be mediating these effects. And in fact, it's kind of incredible to be here because this work very early on was motivated by the studies, the papers of Wazak et al. 2015 Cell that were led by Bart and Alexander that sort of started identifying epigenetic QTLs over a decade ago now. And we have been thinking about how to put that together in the disease context since then. And again, what we had done in the past was show that a large fraction of disease heritability is in sort of annotated regulatory elements, but now we want to understand the actual polymorphisms that are driving it. And so now in this work, led by Dennis Krishin, we finally sort of amassed a substantial amount of data in the disease-relevant tissues, and in particular here we were looking at data from the Cancer Genome Atlas from over 400 tumors with germline genotyping together across a number of cancer types that were all epigenomically sequenced by ATAC-seq to measure their chromatin accessibility. And so when we went back to this data, which was in the public domain, and estimated variants that influence this chromatin accessibility, so chrome QTLs or AQTLs here, we see that there's actually a very large number of such variants, and there's basically no diminishing returns in the sample sizes that we're at right now. Even with 400 samples, we're just sort of continuing to increase in detecting these AQTLs. Moreover, when we integrate these AQTLs into the same kind of estimate of genome-wide disease heritability explained, you can see here the bar with AQTLs in red is substantially more enriched for disease heritability. This is now looking at 7 common cancers relative to just general chromatin accessibility, and more enriched than high confidence EQTLs or coding regions from prior data. And so this started kind of nudging us in the direction, again, that these polymorphisms, which change the chromatin accessibility, are very important and may potentially fill out the missing disease mechanisms. And so we took the same kind of TWAS approach where we were building predictors of expression over here and extended it in some clever ways to the accessibility data that we were working with, where we were now building predictors of regulation or chromatin activity. We're not very creative, so we called these RWAS and CWAS, and took the predicted regulatory activity and associated that with the disease phenotype, all using summary level data. And when we do that, I can break down one cancer GWAS for you. So this is a study of breast cancer, which identified 150 total loci. And in this study, initially, about 35 of these loci were also associated with expression through a TWAS analysis. When we now expanded this with this regulatory variation, we see that an additional 72 or so loci, and in fact the majority of this pie, now colocalize or associate with these regulatory variants influencing chromatin accessibility. So we can actually now fill out a large fraction, potentially fill out a large fraction of these mechanisms when we start looking at regulation in addition to expression. And we see the same phenomenon in other well-powered GWAS, and we continue to see this across other cancers as well. And so one thing that this figure kind of starts to beg the question is, what are these loci that don't have a transcriptional or an EQTL association? So these loci down here and over here, where we only see a variant that affects regulation but not expression. We've dug into these a little bit, and what we see is, for example, as you focus in on loci with more conserved regulatory elements, the associations that you get from a regulatory-based analysis increase, but the associations that you get from a TWAS-based analysis actually decrease as you get to more conserved and more complicated loci. And then likewise, this is in prostate cancer, as you focus in on more prostate-specific genes, actually kind of surprisingly, the associations that you get with the TWAS also decrease, whereas the associations that you get using regulatory variation increase. And so this is starting to come together along the lines of other recent work, for example, the paper of Mostafavi et al. In Nature Genetics, this idea that selection may be making it difficult to identify QTLs in these kinds of loci, but the regulatory elements are perhaps have higher power to detect these kinds of effects. And so just to sort of wrap up this first section, I wanted to sort of cover the general idea that we can use integration, including the TWAS methods, to identify disease mechanisms at very large scale. When we tried to quantify this genome-wide, we see that steady state EQTLs appear to mediate about 10% of the disease heritability. But then now as we're folding in and getting more data from epigenetic features, we see a much larger fraction of loci that could potentially be explained by epigenetic activity. And this is all coming together in a model where there is potentially regulatory element priming, so the polymorphism is already starting to affect the accessibility or the transcription factor binding, but the whole system isn't in place yet because the right context hasn't been met, and so the expression effect is not yet observed. And I think this actually raises some really interesting questions about what does it mean for a variant or a disease association to be functional now that there's a layer of sort of ubiquitous effect and also context-specific effect. In the sort of last part of the talk, I'm gonna switch gears and now talk about work we've been doing more recently that connect germline variation to somatic features and treatment outcomes in cancer. And so as we've seen in some of the previous studies, the GWAS of disease has now grown into the millions of samples, but the GWAS of treatment response, which we're also very interested to understand the mechanisms of, has often been much, much smaller with studies, a lot of published studies merely in the hundreds. And so there's a major sort of sample size bottleneck to moving from case control to outcome. And one potential way to overcome that bottleneck in the world of cancer is to leverage the very extensive amount of tumor sequencing that happens, or clinical sequencing that happens, primarily to identify drivers for targeted therapies for individuals. And this is just showing our institution, Dana-Farber, which over the past decade or so has sequenced 36,000 tumors. All of these are from patients and linked to their electronic health records and clinical data. And so I was very excited, and in fact, at the time that I was considering faculty positions, this is a major driver of my thinking about moving more into cancer and into the clinical space. And then it was sort of only actually after I started my lab that I realized that when people were saying we're sequencing 30,000 tumors, they really were talking about only tumors. This is tumor-only sequencing, and I, as a germline geneticist, actually kind of had no place immediately to fit into this data, and I spent some time in my office sort of silently screaming and wondering what I'm gonna do. And then I, again, came back to one of these lessons, in fact, for my PhD, that we are all much more genetically related than we think, and in particular, this work, I think from 2012, from Bogdan Pisaniak, that in fact, you can use extremely low coverage and noisy sequencing data when you bring it together across lots of individuals to do fairly reasonable imputation of common genetic variation. And so as a kind of last-ditch effort, we attempted to apply this approach to the tumor sequencing data that was being collected clinically. The basic idea here is that when you sequence a tumor on a targeted panel, you're mostly going after these really high coverage targeted exomes, but you're actually also getting a pretty good amount of the read coverage landing elsewhere in the genome in this ultra low coverage off-target sequencing, and most of that or all of that essentially gets thrown away as part of the QC process. But when you integrate the fact that this individual at the germline level is related to a whole population of individuals for whom we do have reference data, then you can actually use these very, very low coverage probabilistic estimates together with the reference data to impute both these polymorphisms and also the polymorphisms you haven't seen along the same haplotype as that individual. And it's kind of amazing that this actually works quite well. So we had about 800 benchmark individuals that had real germline genotyping on a modern SNP array, and we see a correlation of about 0.84 across the entire genome when compared to the imputation that we do off the tumors. And 0.84 is, very bad for SNP array genotyping, but for this, we're essentially getting common germline variation for free out of these tumors we can now use to run discovery analyses. And so now this cohort has come together to be 36,000 tumors with all of their somatic and clinical data together with about 5 million high-quality common variants that we've imputed. And so as a pilot, we wanted to think about the ways that common germline variation could be influencing treatment outcomes in this population. And we focused specifically on the outcomes related to immunotherapy. And I won't go into the details of immunotherapy too much, but it is sort of one of these treatments where the patient's immune system is activated to fight the tumor. And we reasoned that this was a place because there are host cancer interactions where germline genetics may actually have an important modifying effect. And one of the challenges with immunotherapy is that even though it's been quite successful, in some cases leading to exceptional response, it also leads to a very large number of immune-related adverse events in patients. Sometimes these adverse events become chronic, where these patients essentially now develop a lifelong autoimmune condition because of the treatment. And so again, we went back to one of these lessons and we thought, okay, we're gonna look at these mechanisms genome-wide, and we put together, led by Stefan Groha, a genome-wide association study of patients on immunotherapy at Dana-Farber, again, with these tumor-based imputations, we had about 2,700 patients on immune checkpoint inhibitors and replication cohorts, and a large number of other sequence patients. And when we do all of that and look at associations between every imputed common polymorphism and whether these patients did or did not receive toxicity, or actually we do this in a survival analysis, what we see is indeed there are genome-wide significant associations at multiple loci with developing adverse events to immunotherapy. Most interesting to us was this association at the IL-7 locus. And I'll just kind of breeze through to give you an idea of how we can use all of those tools that I talked about at the beginning to connect these new treatment response variants to their mechanism. So we found this locus. First, we confirmed that this was actually a very large effect association with a hazard ratio of about 2 and nominally significant across a broad range of cancer types. So this seemed to have a really substantial impact on all types of patients. We replicated this association with a collaboration from Ben Fairfax in the UK, and in fact in his single cancer replication cohort, this polymorphism now had an odds ratio of 6 for developing toxicities that necessitated steroids. In fact, 50% of the carriers of this polymorphism, which is just a common SNP, it's a 7% SNP, develop these toxicities. So again, a large, potentially clinically meaningful effect. We went to our toolbox of incorporating eQTL data. We actually didn't see any effect of this polymorphism on the expressed gene, but again, we thought about how we could break down this chain further, and when we looked at the raw sequencing RNA-seq reads from GTEx along this polymorphism, you can see here that the non-carriers are sort of quiet for IL-7 expression over here, but the carriers actually right at a junction introduce a new cryptic exon that was previously not known, and that exon splices into the canonical transcripts of IL-7, and this just sort of gets buried when you put all of that together into the total expression measurement. So we identified a new exon which is really strongly significantly associated with the SNP that we think is the causal one. And so this whole cascade came together, starting from a large, well-powered GWAS and then integrating different pieces of data along the way. And so maybe just in the last couple of minutes, I wanted to talk a little bit about why I think that this continues to be a very exciting time, and the sort of amazing work this sort of way of thinking, I think, is now going to benefit from. So again, thinking along the idea of this kind of cascade, first of all, now with the techniques that we've developed, but also I think with just an enormous amount of effort to collect more complex treatment outcome data, we can start looking at the genetics of an entirely new type of phenotype and look at GWAS of treatment outcomes. Additionally, people are collecting population scale QTL surveys of the epigenome. We kind of had to dig around to put together all of this cancer epigenomic data, but these studies are now getting much larger, and I think we'll continue to be able to fill out the landscape of molecular mechanisms through epigenetic activity. Of course, we eventually want to connect this back to the gene, and that means more data and richer measurements of expression and splicing and other expression-related phenotypes in more context, so we can understand the gap between these two measurements. And then finally, there's a lot of work now on connecting all this through to experimental validation. And because you sort of see the word context in different places here, these experimental approaches are also going to need to be both high throughput and context specific, and that's definitely happening as well. And so all of that put together is just to kind of underscore the ideas that we want to have contextual understanding at the entire cascade of a disease variant, and then we also want to go back and forth between genome-wide parameters of the disease architecture and then these local mechanisms which inform the two. I'll also just give a shout out to a review that we recently wrote with Thule, which covers a lot of these ideas and a lot more, and in part in putting together that review and this talk, again, it just underscored to me what an exciting time it is to be doing genetics right now. And with that, I'd like to thank the folks that led these projects, our collaborators, again, the committee for the award, and take any questions.
A question for Sasha.